I'd love to introduce Dr. Alex Good, who's a postdoctoral researcher in my lab. We started in September. Alex studies coral reefs and oyster reefs and is getting into epizootics and coral restoration in our lab. Went to the University of Delaware for bachelor's degree and master's degree, and then went on to Texas Corpus Christi and graduated in May 2025, so relatively recently. Great resource for students who are interested in the next step. So I encourage you to get in touch with Alex. And Alex is always willing
Yes.
to chat about it.
I love to talk.
So without further ado.
Yes, thanks, Dan. Okay. Hi, everyone. My name's Alex. Like Dan said, I'm a postdoc in his lab. We all started here. Dan's lab moved from LSU in September. So Dan and I and Anushka, our PhD student, all started here in September. This is my first postdoc. So this is also really my first seminar presentation I've given. My defense was a year ago this weekend. So
oh my God.
I'm having a little bit of PTSD from that, but I'm really excited to talk to you today about my research trajectory. Hopefully this is helpful for some students, and hopefully other students, staff, faculty get to know me a little bit better, because I would love to talk with more people about our research ideas, any interests, collaborations we have. Dan and I are in Dana Hall, so yeah, it's been really great so far, and I'm excited to be here. Okay, so diving into my presentation, like I said, I'm presenting some of the research from my PhD, and then I'm also going to talk about my postdoc research here at Stony Brook. And I just have a few of my funding sources from Texas A&M Corpus Christi and from my postdoc NSF and NOAA, just to acknowledge them, of course, as well. Okay, so I grew up on Cape Cod, Massachusetts. My dad is a fisherman. That's me. I won that striped bass fishing tournament when I was 10 years old. And I was, I think, around 8 years old when I told my dad I wanted to be a marine biologist. And he was like, how do you even know what that is? But I've just had a love for the ocean my entire life. I got scuba certified in the Florida Keys as young as I possibly could. I was 12 and I've never looked back. Like Dan said, I got my bachelor's in 2017 from the University of Delaware, the College of Earth, Ocean, and Environment. I had an NSF REU during the summer between my junior and senior year of undergrad, where I was at the New England Aquarium and did a feeding behavioral study on the temperate coral, Astrangia poculata. Not sure if anybody's familiar with that, but they're around these local areas. I also got a fellowship through the Scientista Foundation, and it was a Born Seeker Women in STEM fellowship. So I got to speak at Harvard about my experience as a woman in STEM and my passion for science education. After I graduated from the University of Delaware, I didn't get into graduate school right away, and I became a science teacher, an 8th grade science teacher in Boston at an inner city charter school. So that's really where my passion for education, science communication really started. And as you will see, I've kept that going through my PhD. After I was a teacher, I loved it, but I missed research a lot. So I went back to the University of Delaware and got my master's degree, where I did a study on using PLA, which is a biodegradable filament, to create artificial reef structures and outplant those in different proportions with living corals and test the effects of that structure as a restoration technique. So we looked at fish feeding behavior and coral growth, and all of that work was done in Fiji. I also had the opportunity a few summers ago to go back to Fiji to teach a coral reef conservation course through the Sea Education Association, which is based out of Woods Hole, and got to go back to Fiji and continue my love for teaching. And then I went to Texas A&M, Corpus Christi, got my PhD there. I did some coral work, some oyster work, and created a STEM education curriculum that I'm excited to talk to you about today. So the overarching question for theme for this seminar is, what determines whether a reef survives and who or what can tip that balance. And I'm using sort of three pillars to address this overarching question, looking at species responses, conservation strategies, and education approaches. So I'm going to start with my PhD research, then go into my postdoc, and then end with a few notes about some future work I'm hoping to do. The species response question that I asked during my PhD are the organismal and population level tolerance thresholds of keystone reef species to interacting local and global stressors. I also published a review paper on coral reef conservation strategies under interacting stressors that I'm going to talk about in a moment. And then my education, my high school STEM curriculum that I implemented and tested in Texas to see how experiential learning can influence students' attitudes towards STEM. So starting with the review paper, this is sort of where my PhD and my research really all started, because I started my PhD during COVID. So I had a lot of free time on my hands. I couldn't do any of the research that I had gone there to do. So I just dove right into the literature and I wrote this review paper about the coral reef conservation crisis and this debate about should we be addressing the global climate change issue or should we be addressing local stressors that are degrading coral reefs, and what's sort of the balance between those local and global stressors for conservation. This is the main figure from this paper where I sort of synthesized these science-based solutions that we could use towards conservation. And this was sort of the basis. I was also my advisor's first ever PhD student. So she was like, go do whatever you want to do for your research. And I had a lot of time to think about what I wanted to do. And as I continued thinking about my research plan, I wasn't as interested in assisted evolution techniques. So for the rest of this talk, you're not really going to see anything about assisted evolution. But I was really interested in this idea of risk assessments and predicting tipping points for these coastal ecosystems, and also focus management strategies and integrating this ridge to reef or land to sea conservation approach. So getting into my species responses, I looked at is this echoing? Oh, okay, it's okay. Okay, it's just me. Okay. I looked at the tolerance threshold of both corals and oysters. And you might be thinking, why did you do corals and oysters? Well, they are both very important keystone species. They're ecologically and economically important reef structures for habitat, biodiversity, coastal protection, and fisheries. There are some key differences between coral reefs and oyster reefs, such as corals form different morphologies. They're found in usually warm, shallow, clear tropical waters. They have a much slower growth than oysters do, and they're especially important for the tourism industry specifically. Whereas oysters improve water quality through filtration. They are in a much broader range of estuarine conditions. They have a much faster growth, and they're especially important for the food industry. So how did I measure the tolerance thresholds of these corals and oysters? I did this two ways to measure their physiological tolerance. First, I measured the metabolic oxygen levels, and I did this through a technique called intermittent flow respirometry. This is very similar to going to the doctor and doing a stress test on your heart, where the doctor might tell you to walk and then run and then walk again, and they're going to see how your heart rate fluctuates with that change in stress. And this approach is very similar to that in if you have time on your x-axis and you have percent oxygen saturation on your y-axis, the animal goes in the chamber and there's a flush pump that is on, bringing 100% oxygenated water into the chamber. After a set amount of time, you turn that pump off, and now you have a closed chamber system. And that green box highlighting the letter C is showing that's the rate of change of oxygen consumption that you measure over a set period of time to get this metabolic oxygen number. Once the oxygen saturation in the chamber dips to about 80%, you then turn the pump back on and oxygenated water fills the chamber, and you can repeat this cycle over and over. This is really great technique because you don't get waste products built up in the chamber. You can measure the physiological response continuously over time. And it's usually done on fish. So if you've heard of this before, you've probably seen it done from some fish physiologists, but I decided to try it out with corals and oysters. The second variable I measured was net calcification. So both corals and oysters are calcifying reef-building organisms. And I used the total alkalinity anomaly technique to measure calcification. And so what this tells us is you measure change in total alkalinity between the sump water and the chamber water, and how different a 2 molar change in total alkalinity is telling you that there is a 1 molar change in the formation or dissolution of calcium carbonate. Basically tells you how fast or how much they're growing or dissolving. Okay, so I'm going to get into the oyster work. I'm pretty sure a lot of people here are familiar with the Eastern oyster since we're in New York, but I don't know how familiar you are with the Eastern oyster in Texas. So there is these genetically divided populations of the Eastern oyster in Texas, which is super interesting and still not very well understood, because you'll see here these pie charts are a breakdown of the genetic configuration of these two populations. And you'll see right in Corpus Christi Bay, where I was at school, highlighted by the red box, there is this divide in the genetic makeup of these oyster populations. They also don't really hybridize in Corpus Christi Bay, and scientists, we still don't really understand why there are these genetically different populations in this area. And I will note the white pie charts on the northern part of the Texas coast, that's about 98% similar to the rest of the Eastern oyster all the way up the East Coast into Canada. So for some reason, Corpus Christi Bay south, the Eastern oyster genetics are different. So one hypothesis we have might be the different environmental variables along the Texas coast. Texas has a very interesting environmental gradient to salinity and temperature. So in the north part of the Texas coast, up near Galveston, you have very, very freshwater-based systems. The salinity can drop down to 2, whereas down in the Laguna Madre estuary in the southern part of Texas going towards Mexico, the salinity can get up to 50. So it's really, really hypersaline down in those areas, those estuaries. So one hypothesis is that maybe this salinity gradient is driving the genetic differences. But you also do see differences in summer temperatures based off of where you are along the coast. So the southern part of the Texas coast stays hotter for a lot longer of the year than the northern areas do. So to test this, I looked at the Eastern oyster, the 2 different populations. I looked at the metabolic responses of metabolic oxygen and calcification. And I looked at the interaction between this local stress of salinity and this global stress of high temperature. And this is just a photo of what my experimental setup looked like. And that's an oyster in a chamber all the way to the right. You will notice that, well, I don't know if you noticed, but I have video cameras watching the oysters. And you might be thinking, why are you videotaping the oysters? They don't move. They're not very exciting. Well, they do open their valves. They do gape. And this makes it very challenging to better measure their respiration rates, because if they want to stay closed and not breathe, then they can just stay closed and not breathe. So that made it very challenging for me when I stressed them out. They would often just close up and not open, and I had no data. So this is very important to note because it's a big difference between the corals and oysters. Corals are always breathing or producing oxygen, which I will get to, and oysters are not. So this is sort of what the intermittent flow respirometry data looked like. Each of those numbers is a different cycle. And you can see, for example, the difference between cycle 4 and cycle 5. In cycle 4, the oyster is clearly closed, not breathing at all. There's no change in the oxygen levels in the chamber. Whereas in cycle 5, the oyster probably opened and was filter feeding, breathing a lot. So this is important to note. Getting into some of the results, we did see a higher metabolic oxygen rate in smaller oysters. So we have weight of the oyster or biomass on the x-axis than in the larger oysters. This is really important because this research was funded by Texas Sea Grant to support the aquaculture industry. I was at a local hatchery actually doing this work. So the fact that the smaller oysters are opening more and breathing more is actually a good sign. So breathing more for oysters doesn't necessarily mean they're stressed or that there's a negative impact. It actually means that they're opening, they're feeding, they're healthy, because when they're stressed, they would close. So a lot of these larger oysters, as you can see, the metabolic oxygen rates were very close to zero. So they were staying closed a lot of the time. So when we look at the interaction between temperature and salinity, you will see another one of these figures. So just to orient you to it, you have your local stress of salinity on the x-axis, where you have your low salinity values, your control salinity values, and your high salinity values. And then the black and red are the global temperature stress. So black is the control temperature and red is the heated temperature. And what we see is that salinity is really driving the change in the metabolic oxygen levels, whereas in the control salinity, you're having your highest metabolic oxygen respiration rates. And at low and high salinity, those rates are a lot lower and the oyster is staying closed. However, we do not see a synergistic effect of these two variables. We actually see an antagonistic effect where really, again, that salinity is driving most of the differences. Another interesting thing we found is we did see a significant correlation between whether the oyster was breathing and calcifying or dissolving its shell. So when the oyster was open and breathing, there was higher dissolution rates, versus when the metabolic oxygen levels were lower, there was higher calcification rates. So this is an interesting trade-off between the physiological parameters that we're seeing with these oysters. So that's interesting to know. So just to wrap up the oyster results, we didn't see any differences in the thresholds between the north and south, which was surprising for us because we were thinking maybe that was driving some of the genetic differences. We did see metabolic trade-offs by treatment and size, but salinity was the primary driver of this difference and acted antagonistically with high temperatures. So getting into corals are a little bit different, or very different in some ways, than oysters, because they have a symbiotic relationship with an algae called zooxanthellae. And this symbiosis accounts for 90% of the energy that the coral needs to survive. So this is highlighting a little coral polyp. Corals are colonial animals made up of thousands of polyps. And inside the polyp, inside the coral tissue, you can see those brown-green dots. Those are the symbiotic algae that are producing photosynthesis and doing photosynthesis and producing the energy that the coral needs to survive. We also know that there are various local and global threats threatening coral reefs across the globe. Locally, terrigenous-based sediment is coming from the shore and suffocating the corals, basically, where the corals are unable to feed with their tentacles, and they're also limited in their light availability to do photosynthesis from that turbidity and sediment. And then, of course, you might have heard of coral bleaching, where the high temperatures are causing a breakdown in that symbiosis between the coral and its algae. So these are the two local and global stressors that I looked at for corals. We do also know with corals that there is this ridge to reef impact, which brings us back to the terrigenous-based sediments. So when this study showed that if we have our watershed health index on our x-axis and we have our reef health index on our y-axis, when you have a healthier watershed on land, you have a healthier coral reef in your coastal waters. So there is this significant correlation. And this is especially important in Hawaii, where I did this research, because you can see the variability in the watershed health indices around Oahu. The warmer colors are the less healthy watersheds. So that's right around Honolulu and Waikiki Beach and a lot of the popular tourist areas. And then the cooler colors are the healthier watersheds, where there's less coastal development and just less pollution from the watershed. I did this work in Kaneohe Bay, Hawaii, and Why I'm highlighting the bay here is that I also looked at two different locations of corals in the bay. This southern part of the bay, Bokaloi, or where the Hawaii Institute of Marine Biology is, where I did the work, that's in the southern part of the bay where you see a less healthy watershed. And that's because the military base was built there. There's a lot more houses around that part of the bay. There's also not as great water movement through the bay there. So there's a longer residence time of the water in that part of the bay. So the temperatures stay warmer there and pollution isn't able to escape as easily. Whereas in Hatchery 44, the northern part of the bay, you have a much healthier watershed because there's a lot less development in that area. And you can see these corals are in contact with the open ocean there. So they're really getting a lot of those cool ocean waters coming in consistently. So I collected corals from both of these parts of the bay to look to see if there's differences there. So similar to the oysters, this is my experimental setup in Hawaii. I had corals from the two different locations. I tested the coral host response to turbidity and temperature. And then I also tested the photobiology response of that symbiotic algae to turbidity and temperature. And that's sort of what the turbidity treatment looked like, the far chamber on the right. And what we found on the coral host is that temperature and turbidity had a synergistic effect on the respiration rates of the coral. Or I should really say coral host, coral holobiont. Because what's different here than the oysters, and that's why I have this 0 line here, is when you have a positive respiration rate, that means the coral is doing photosynthesis. So that means the algae is healthy, photosynthesis is happening, we're creating oxygen in the chamber. If you have a negative value, that means respiration is happening. So this photosynthesis to respiration ratio is off, where the coral animal is breathing more than the algae is producing oxygen. So this would be a sign of stress if you have a negative metabolic oxygen rate. So you can see again, we have our local stressor on our x-axis, turbidity and temperature, high temperature in red. And we can see this synergistic decline under high turbidity and high temperature in the metabolic oxygen. And just looking at the 4 treatments there in the second graph, you can see the significant decline of respiration under high temperature and that interaction treatment. How I measured the health of the symbiotic algae is through pulse amplitude modulated fluorometry, otherwise known as PAM. What PAM does, you can see a picture of her there. She flashes a series of actinic red light at the coral, and that measures the productivity of photosystem 2, where photosynthesis is happening. So the values of FV over FM is the photosynthetic efficiency of those photosystems, so the productivity of the algae to take in light and create energy. And then the non-photochemical quenching is a stress response. So it's basically telling us how much heat the algae is taking in, and then, or how much light the algae is taking in and dissipating as heat versus using it as for photosynthesis. So it's a stress response, protective mechanism. So just to start with how many symbionts these corals even had, I want to note that the blue is the HIMB corals, and the gray are the Patry 44 northern corals. And right away, we saw an overall significant difference between just the number of symbionts that these corals had. So the HIMB corals had significantly more symbionts, which means if you have more symbionts, we would assume you would be more productive. But what we saw is actually the opposite, where there was a significant decline in the photosynthetic efficiency of the HIMB symbionts compared to the Patry 44 symbionts. If you look at the gray boxes across the treatments for FV over FM, it stays pretty consistent, whereas the HIMB symbionts significantly declined under that high temperature and interaction treatment. So just because you have more symbionts doesn't mean that you're actually more efficient at doing photosynthesis. Same trend here with the HIMB corals. When we saw the decline in photosynthetic efficiency, we saw an increase in that non-photochemical protective mechanism. So this is telling us that as the temperature increases, that those symbionts are dissipating that energy as heat, as a stress response. So in conclusion, we did not see any differences in the metabolic oxygen thresholds between locations, but we did see a synergistic effect of temperature and turbidity on the metabolic oxygen and a significant decline in photobiology, but just for the HIMB corals that was driven by high temperature. And just in summary, to wrap up this species response of that I did for my PhD, if we look at the oyster tolerance thresholds, we have our local stress of salinity on our x-axis and our temperature stress in red. And same for the corals. We have turbidity, the local stress on the x-axis. What I want you to note here is that what I took away from this is that local stress is playing a significant role in the acute metabolic suppression of both of these keystone species. So yes, temperature stress is a problem. It is affecting them, but really the primary drivers from both of these experiments were in these local stressors. And just to wrap up my dissertation work, I want to talk to you about the high school education curriculum that I created. I asked the question about experiential learning influencing student attitudes towards science in the classroom, STEM careers, and environmental literacy. Why did I do this? This might not shock any of you, but there was a press release in 2013 that said Texas is last in ranking from all the United States on ocean protection. Shocker. And when you make a report card of the health of the bays along the Texas coast, you can see that really none of the bays along the Texas coast had healthy watersheds, so to say. And this was a report done by the Heart Research Institute where I worked. So basically what I want you to know from this is that Texas has many important very, very important coastal ecosystems. I could talk about all of them, but what Texas needs is environmental stewardship and environmental literacy to help them protect these coastal environments. So I brought the STEM curriculum to the West Oso High School in Corpus Christi. I want to note here that this is the red arrow is showing you how close the school is from the coastline and from the beach. That Cole Park is a very pretty beach. And most of the students, when I ask them, say they have never actually been to the beach. So this is a Title I school district with very limited resources. And these students really do not have the opportunity to be out on the beach or enjoy the ocean much at all. I worked with the 9th grade students. I worked in 2 teachers' classrooms, 8 classes total. I surveyed 100 out of 130 students. Those other 30 students either didn't want to participate or were in school detention or something like that. 44 of the students were females and 56 of the students were males. So the research question was kind of broken down into 4 categories: the students' attitude and self-efficacy, how they feel about themselves and their science skills, their interest in having a STEM career, and their attitude about environmental protection, and then specifically their knowledge about coral reef ecosystems. What I did here is I broke up the curriculum into 3 different teaching styles: a traditional classroom lecture, hybrid learning with some virtual and hands-on laboratory work, and then a field trip out to the beach to actually do a beach cleanup. I looked at the difference in responses between males and females, and then also the survey questions were broken up into these 4 categories, which I'll show you in a moment. So this is just sort of a breakdown of how the curriculum went. So think of this as like day 1 on the left all the way to the end. I was there in the classroom for about 2 weeks. What I did first is I gave the students the survey before I even got there. So they hadn't met me, or this was just sort of the control of the experiment, their baseline knowledge. Then we did the traditional lecture. We learned about coral reefs, their biodiversity, ecological importance. And this had like the 3 main sections were coral biology, coral ecology, and then impacts and solutions to help coral reefs. I will also note this traditional lecture followed the Texas education standards so that teachers can actually implement it into their curriculum and use it towards their state testing requirements. most people probably know these as next gen education standards, but in typical Texas fashion, they have their own education standards. So we followed those for Texas teachers. Then after that lecture, the students took the same survey again. After survey number 2, we did a virtual field trip to the Hawaii Institute of Marine Biology that was hosted through the Texas State Aquarium. The students basically could watch videos and interact with my advisor, Dr. Barr, as she was doing this ocean acidification experiment at the Hawaii Institute of Marine Biology. I also interviewed all different types of marine scientists from diverse backgrounds to try to get the students to learn more about scientists that looked like them and came from places that they came from. And we watched Chasing Coral, which if you haven't watched it yet, highly recommend. After that, we did the hands-on lab activity where I actually brought back corals from the ocean acidification experiment, and the students measured the changes in the growth of those corals based off the different treatments that they were in. So the students learned about ocean acidification and created this lab report about the results about coral growth. Then they took the survey again, and then we went to the beach clean up. We cleaned the beach. They also got to go to the rescue center at the Texas State Aquarium, which they really enjoyed, where they rescue a lot of marine mammals and sea turtles. And then they took the survey for a final 4th time. This is sort of what the survey looked like. It's broken up into those 4 categories, and each question was a Likert scale, 1 to 5, strongly disagree to strongly agree. And we asked questions like, science is fun. I see myself as good at science. I would like to have a career in science. Coral reefs are important, things like that. And I have lots of figures from this, but just for the sake of time, I just took like the main total score from the surveys over time. So I did break down the surveys into each of those categories to look at how the student responses changed by survey category. But this is just showing you just the mean total score of the survey. So you could say the survey was out of 140 points, which is the gray dotted line at the top. And then the x-axis has your different survey time points. So your control, your traditional lecture, your hybrid learning, and your field trip. And when all males and females were pooled together, we saw a significant increase in the student score after the hybrid learning, and then another significant jump in their score after the field trip. And we didn't really see a change in their answers based off the traditional lecture. But then breaking it down by males and females, females in pink, males in blue, we did see that that jump after the hybrid learning was specifically from the female students, and the male students didn't increase their scores until after the field trip. And so just to give you like a quick overview of the rest of the results, we did see a significant increase after the hybrid learning and the field trip. Females had the highest increase in their scores, and this was particularly in the career and environmental categories. The male students never increased their score in the STEM career category, which I found very interesting. And the coral section had the highest increase in scores overall, which is not surprising. Those were the general questions like, I know what a coral is. Corals are important for biodiversity. And that was the largest jump in overall score. I just wanted to end this section with just some of the quotes that I got from the students, just because I think this is really great to see. One student said, no one in my family has gone to college yet, so I want to be different and I won't let anything hold me back. It was fun to use my brain to be creative, picking up trash and making art out of it and meeting the scientists. I think we should stop littering and people should be in jail if they litter. And then there won't be pollution and it will be a nicer place to live. The lab was my favorite part. It was fun. We're making a change when we were at the beach picking up trash. And then it's important to protect coral reefs. It might not seem important now, but we will see the effects later if we don't do something now. Okay, so that was an overview of my dissertation, which is kind of a lot, but I quickly do want to talk about my postdoctoral work and the Seascape Ecology Lab, some of the things that we're working on. I don't have results yet starting in September, but I do want to go over sort of our approach, some of the research questions we're asking. Again, my postdoctoral work sort of breaks down into these three pillars as well. Starting with the species responses, I'm part of this PREDICT team funded by NSF, which is a multidisciplinary group of very impressive scientists from 4 different institutions. So we have microbiologists, immunologists, modelers, which is our lab, ecologists, and we're all working together to understand stony coral tissue loss disease dynamics in the US Virgin Islands. And my specific question is trying to understand the heterogeneity of the coral communities and how that impacts the severity of the outbreak of the disease. And so why I'm considering this, I'm really modeling the difference in disease spread across the island, the different bay systems. But the real driver of this question is the difference in the coral communities and the different tolerances that these species have to the disease. Then for conservation and education, I'm also working, 25% of my work is based off this NOAA CMR3 grant, where I am doing some connectivity modeling, but that is to understand coral larval dispersal based off hydrodynamics and see where the optimal coral reef restoration sites would be around the US Virgin Islands. So this is directly to impact conservation and management. And with this NOAA grant, we have a lot of community partnerships and education work going on St. Thomas, which is really great. So just to get into stony coral tissue loss disease a little bit, I don't know if you've heard of it, but it started in the Port of Miami in 2014 and is the most severe coral disease in history. And it started in 2014, and then all the way through 2023, it's been documented spreading down the Florida Keys reef tract and all through the Mexico Caribbean reef systems. So it's a very deadly epidemic for the coral reefs. And what makes this even more complicated is that there are 20 plus species of coral that are affected by this disease, and they all respond to this disease in a different way. So this is if you're scuba diving or snorkeling on a reef, this is what SCTLD might look like, where the white areas are the diseased tissue versus the healthy tissue. And not to be confused with coral bleaching. So with coral bleaching, really most of the colony will turn white, whereas with the disease, you see more lesions or spots of the disease. So the data that I am working on was collected in US Virgin Islands from 2019 to 2024. So when the SCTLD first got to the Virgin Islands in 2019, and we have preliminary data of these bay systems before the disease outbreak began. So we have some preliminary baseline data. And then our team was boots on the ground. They were in the water as often as they could be, surveying these reef systems as the disease progressed. You'll also notice the little needles. Those are spots where teams actually deployed antibiotic treatments to the corals to try to help slow down the spread of the disease. But for my modeling right now, I'm not looking at the antibiotic responses. I'm just looking at the areas where there was no antibiotics deployed. So what our team did was they did these radial transect surveys. So they found a Colpophilia natans colony on the reef, and they kind of made that their starting point for the survey. They tagged it so they could go back to that same colony over time. And then they swam in 1 meter intervals in a circle around this center colony. And while they were swimming around, they were counting how many corals had disease and what stage of the disease the coral was at. So we can take the original species counts from these bays, and we can take the health surveys, and we can calculate disease prevalence, the number of diseased corals versus the total number that are susceptible to the disease. And what we can do from this, or what I hope to do from this, is create these SIR models. This stands for susceptible, infected, and removed. This is a classic disease epidemiological model that's been used for hundreds of years to study disease spread in humans, for example, COVID-19. And what this model tells us is that you have a group of susceptible individuals in black, that black line on the graph. So say that starts at 100%, everyone's susceptible before the outbreak happens. And then you have an infection rate of beta. So the number of people that are susceptible that then get infected in blue. So you see that peak in infection rate. And then as the infection rate declines, that's because the individuals are either recovering or, in the corals case, removed, because a lot of corals don't survive this disease, unfortunately. And then that's where we get our gamma removal rate. So I'm not going to read through all of this for the sake of time, but what I want you to know here is that infection rate that I was talking about, the susceptible to infected, that's different for every single species. So this is sort of where the species response comes in. You have high susceptible corals, moderate susceptible corals, and then low susceptible corals. The high susceptible corals get the disease right away and die very quickly. Whereas the low susceptible corals might take a while to get it, they'll hold on to it for a while, and they'll keep infecting all the other corals around them. So this disease pool, or the difference in susceptibility, is really where my modeling comes in. And this is important because all of the different bays across St. Thomas and St. John that we surveyed have significant differences in their community composition. And this is just one of the sites where this first bin here is the susceptible corals by susceptibility group. So the pink are the high susceptible corals, yellow is moderate, blue is low susceptibility, and you can see the change from susceptible to infected with SCTLD to then dead. And really what I want you to take away here is that in the dead bin, all the way to the right, those are all high susceptible corals that we have documented to have died. The moderate and low susceptibility corals never really fully died. Their whole colony didn't die. And then I made some quick SIR models to show you, broken up by susceptibility group. And you can see here that the high susceptibility group really shows a good trend of decline of susceptibility, peak in infection, and increase in death. Whereas the other moderate and low susceptibility corals aren't really showing that trend. And this is probably because corals aren't humans, right? Corals are colonial animals. So a few polyps, a few might die, but if the whole colony doesn't die, then we're not considering it dead. So there are some caveats to this data that we are trying to figure out to understand the spread of the disease. And last but not least, for the NOAA grant, I just wanted to show you a quick photo of sort of what our larval dispersal looks like. So this is supposed to simulate if we did a restoration project in Lancashire Bay and we restored a bunch of corals there, and then those corals reproduce, where will their larvae spread based off hydrodynamic data that we got from Woods Hole Oceanographic Institution. So the red dots are showing those larvae only responding to currents. And then the blue are if you incorporate currents and wave into larval dispersal. So we're starting this now. Haven't dove into this as much. But this is really important because Lancashire Bay is a National Park Service restoration site for corals. And we're also doing this in Flat Cay, which is a territorial priority for coral reef restoration. And then the education aspect of this is I've been helping a master's student at the University of the Virgin Islands, Khalifa, do her corals in the classroom. So similar to my STEM education curriculum, she is bringing corals to classrooms on St. Thomas and teaching the kids all about coral biology and disease and reproduction. So Khalifa and I actually met at a conference before I even started working with Dan and before I even got put on this grant, and we connected beforehand because I presented my education chapter. So now it's really great full circle that we get to work together. And just in the future, some questions that I hope to ask are looking more at the larval behavior and survival and settlement. So maybe getting back into that physiology work that I did during my PhD and trying to understand what that physiology of the coral larvae would have on reef connectivity and resilience. And then with that, how do we connect more people to this data? The public, resource managers, the reproduction lifestyle, the coral larvae stage of the coral lifestyle is an invisible stage. Not many people really see it or understand it, but it's really what's driving reef recovery and resilience. So it's something that I'm interested in trying to educate more people on. And with that, I'll take any questions.
So we, for two students,
Okay.
first few questions, I think Kayla and Jacqueline are two students. I can go first. Thank you for your talk. It was really interesting. My question was related to your dissertation work. You had focused more on like, or trying to answer why, what's it, the Eastern oyster population called like genetic variation that and again more like abiotic environmental variables. But like how much did you consider or like do you think that a more density dependent regulation has an effect on it, like competition?
That's a good question.
Disease.
Yeah, that's a great.
Can you repeat the question?
Oh, yeah. So the question was that I looked at abiotic factors, but you're asking more about like community factors like density dependence or competition and how that might impact how the oyster responds on a reef, right? Okay. That's a great question. I tried to fit a lot into this short seminar, which maybe is a flaw, but what I didn't say is that this work was mostly aimed towards the aquaculture industry, so not natural oyster reef populations. I mean, it could be applicable to that, but what we did is I actually started from the beginning of spawning these larvae in the hatchery that are then going to be, the seed was going to be distributed to farmers along the Texas coast. And this was because the aquaculture industry in Texas had just started when I moved to Texas in 2020. It's the last coastal state, I think, in the US to get an aquaculture industry. So I didn't consider ecological factors because of that, because I was thinking of more growing them on a farm in a cage. But if I had to speak towards that, there is a lot of variability in density in the natural oyster reef populations along the coast. Some are much more overfished than others. And I do think that that would be something to consider if you were collecting oysters from a reef that had a lot of oysters and one that didn't. Maybe you would see some differences in their behavior. Yeah, of course. Also competition, because if they're threatened, they stay closed. So the predator response would also happen there if there was a lot of predators.
Thank you so much for your talk. It was really, really interesting to learn about.
Yeah.
I was really interested in the outreach work you did because that's where conservation really starts. And I was wondering if throughout your research, if you found why there might have been like disparity, especially amongst genders in STEM education and interest, and if there's any approaches you can do to bridge that gap.
Yeah, so a lot of the preliminary research that I had read was that females had a really strong interest in STEM until middle school. And then after middle school, it dropped off. And there was a lot of studies that showed that that could be due to a lack of like confidence or seeing themselves in that field and really picturing themselves doing that. And then they get to high school and the interest in STEM sort of declined. So that's why I wanted to do it with high school students, because I wanted to try to understand that difference between males and females and at the high school level. Because also a lot of studies are done on elementary school kids. There's not a lot of education studies done on high school kids, maybe because they can be difficult. Which I will also note, I had to go QA/QC my survey responses very detailed, because like in a very detailed manner, because some students just selected one for every answer and those were taken out because they're just some stubborn. It was the end of the school year also, so we have to consider that. They were ready for summer vacation. But the students that were interested in the survey, and maybe that's it too, right, is that females were a little bit more interested in me being there. Some people have asked me, do you think you being a woman and speaking to them resonated more with them? There's a possibility. It's hard to really break down. I did interview the students, but again, the interviews are sort of hard to, that qualitative analysis is sort of hard to really parse out what the drivers are. So yeah, it's really hard to tell, but I think that it is interesting that we did see that increase when we were directing it directly at the female students. So, yeah. Yeah.
I think Trinity.
So regarding the same STEM education topic, I was wondering if you looked into anything other than the interviews and the Likert survey data. Like, did you specifically look into if they like went into STEM fields in college?
Yeah, that's a great question. So it's they were freshmen 2 years ago, so I guess they're only juniors now. My collaborators at the Texas State Aquarium and at Texas A&M Corpus Christi have done a similar project to that, where they have a summer camp at the Texas State Aquarium and they watched where those students went through the years. And a lot of those students did end up in marine bio programs at A&M Corpus Christi. So there is an interest in following the career progression of these students and how these programs, like the one that they have at the Texas State Aquarium, how those programs might help those students into the future. So I didn't specifically do that, but it is happening. Yeah.
Thank you.
Trinity.
Thank you for your talk.
Yeah.
I was interested, you mentioned that your lectures, like the lecture section of your high school education, was like in line with Texas's standards. What does that entail exactly? Do you think it supports their education, or like did you find difficulty working with it? Could you speak more on that?
Yeah, definitely. Just logistically, that work, the curriculum is in review right now. So the hope is that it gets published. It's then a published curriculum that any teachers could use towards their teaching standards. So since I was in Texas, I decided to use those standards. But any teacher could use this curriculum. I just decided to base it towards that. It was also more motivation for the teachers to have me into the classroom because I was basically creating a free curriculum for them to use continuously. And the standard that I chose was like the ecology standard. So about like symbioses and community composition, trophic food webs, carbon cycling. I chose that portion of its general biology for ninth graders, and that was the education standard that I chose. In terms of differences between we call them TEKS and the next gen, there are some challenges. You cannot use the word climate change. You cannot talk about evolution, really. There are definitely some barriers. The teacher I worked with didn't believe in climate change, so I was teaching ocean acidification. So, well, I shouldn't say she didn't believe in climate change. She didn't think that humans had an impact on climate change. She didn't think it was a human-induced effect. She just thought that the ocean's acidification was changing because of natural causes. So, yes, there were challenges. Yeah, I don't. Yeah.
I'm thinking in terms of a natural pattern of education for a person. They might go out to nature and observe things directly. In other words, do your field portion first in their lives and then go into a classroom. So I'm wondering if you had reversed the sequence of exposure, if you think you would have found the same pattern.
Yeah. I do think that there definitely would have been more of an interest probably in the lecture or even the lab if they had seen the beach first. I did have that proposed to me where so I had 8 classes of students. They were like, why don't you just take a few of the classes to the beach first? Mix the order around, like you're saying. Unfortunately, my resources and the time that I had, it would have made it like logistically very difficult for me to do that. But that was something that we are very interested in. And I think future studies should incorporate that change. Because there's something to be said, like maybe at the beginning of the school year, if you're a science teacher, you do something hands-on or out in nature right away and really hook your students. I think there's definitely something to be said for that. Yeah.
One thing I've observed with undergraduates here at Stony Brook is that if they know what the other students in the class are going to say, they all say the same thing. So that if you ask a question out loud to a class, why did you take this class? Every single person in the class will say the same. So I wondered whether these kids knew what each other was saying, how much interaction there were between students.
Yeah.
And because there is a great, I think I was involved for a while with WISE here at Stony Brook. And one of the big issues is whether the decrease in women involvement in STEM is related to the peer pressure of not wanting to look like you're too smart or not look, embarrassing yourself in front of boys or whatever. I mean, I had to say, it's upsetting to me that this is true, but I haven't observed it.
Yeah. I totally agree with you. And to speak on that, the interviews I did in private with just me and the student. So I definitely got more vulnerability and honesty from those interviews, like some of those quotes I showed you. Again, I'm still trying to figure out a way if there is a way I could do a qualitative analysis. Unfortunately, I consider myself more of an ecologist than an education researcher. So I'm not very familiar with a lot of qualitative. I just did basic stats on the Likert surveys. So I don't consider.
You can't have them do the survey orally. It's what a part of, like, they do a survey, if you have them do, they.
And they say it out loud.
The question.
Yeah, the survey was on their own laptop. They couldn't see each other's answers. They were on their own laptop. They couldn't talk to each other. I will say the teacher, there was some differences between me and the teacher, but they were very, very good teachers. So I had the support of the teachers where if there were students that were maybe thought distracting, they separated students, they didn't put friends together, they were at separate tables. So I do think I avoided that, what you're talking about, as much as possible, but the only time they really talked to each other and worked together was during the lab. They all really liked the lab because also, I forgot to mention this, but we put oysters shells in vinegar and then the next day came back and weighed them and they loved the mushy oyster shell after being in the acidic vinegar. So that's a fun one if you're ever with kids. But, yeah.
Hi, I really liked your lecture. I'm curious, I know that we talked earlier about how currently the age that the students are at, we can't really look at their career trajectories. But I am curious to know if there is any interest on your part or any of your collaborators in seeing how perhaps for distributing this survey again, several years later and seeing it how long these interests last for, or do you know what I mean?
Yeah. So
only the time they were doing this, a year later, will they still feel as strong?
Or what they remember even about me.
Yeah.
Do they remember me? Great question. I did go back the next year. I went to the school every year for career day and spoke for career day. And I did have some students come up to me and been like, I loved your coral reef curriculum. Most of them were females. So I think that would be really cool. We still have the surveys. Obviously, the teachers that I worked with are still both there. It was also great too, I forgot to mention this, that one of the teachers was from Mexico. She spoke Spanish fluently, and a few of the students only spoke Spanish. So I did make the curriculum in both English and Spanish, and she was able to translate it for me because I do not speak Spanish. But that is something also. So the teachers are still there, and the students would be seniors soon. So that is something to maybe consider. I could give her the survey. The only thing is they kind of break, they're not in the same classes, and
They're not taking biology.
and they're not taking biology. And I think they only have to take, I don't think senior year they have to take a science course. I think they can opt out after they take, I think they take a general chemistry sophomore year, and so it gets tricky, but it is interesting.
Yeah, I don't know, I don't know the school, I don't know Texas education well, but I feel there is definitely a way to do it, even if it's a little logistically difficult.
Yeah. You probably have this, probably have to have the students opt in to do it after school or something, because they're not in the same class groups and things. So
yeah, I don't know, maybe during homework. I'm just curious because when we're talking, it seems you're getting at this idea of sense of belonging and stuff.
Mm-hmm.
And my work with that has been targeting young women.
Mm-hmm.
And I've also done some research, and women and students of color are more likely to feel like they are the type of person who belongs in STEM. And so I'm just curious to see if this was a foundational experience for them, or if after a year afterwards, this is no longer something that was resonating with them.
Yeah. So maybe if it didn't resonate longer term, then maybe a different approach would be better. Yeah, I think that would be really, really cool. And I still am in contact and collaborate with the people there. So the teachers and the people at Texas A&M. So it's definitely something we could consider. And I really love that idea. I will say too, that the virtual interviews of the scientists that look like them, which were a lot of women and people of color, I do have like a public YouTube series of those interviews. So if anybody's ever doing education with like minority groups and you want to view some of those interviews, they are like publicly available. So it just asks the scientists about where they're from, what their research is, why they got into this field, their favorite part and their most challenging part of being in the field. So there's scientists from all over, Puerto Rico, Nicaragua, Mexico, yeah.
Thank you.
Yeah, Texas.
Well, we have lunch for everyone. So thank you, Dr. Good.